Cloning of antibiotic resistant gene in E. coli. So the ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. So this is the cloning. The ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. Now tools of recombinant DNA technology involves first restriction enzymes. It is restriction endonuclease. Now one of these added methyl groups to DNA while the other cut DNA. The latter was called restriction endonuclease. Now, what is that? The recognition sequence. Now, this specific base sequence is known as the recognition sequence for HIND2. Now, what is that? It was found that HIND2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. So this specific base sequence is known as the recognition sequence for HIND2. Okay, now in the second, like restriction enzymes belonging to a larger class of enzymes called nucleases. Now, so these are of two kinds, so exonucleases and endonucleases. So there are nucleases that restriction enzymes belongs to kinds of two, first exonucleases and endonucleases. Now there is palindromic nucleotide sequence. So each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Example, GAATTC recognized by EcoRI. So GAATTC, you see in the diagram like so. Now a steps in formation of recombinant DNA by action of restriction endonuclease enzyme EcoRI. Steps in the formation of recombinant DNA by action of restriction endonuclease enzyme EcoRI. So you see in the diagram, so addition of restriction enzymes. So the enzyme comes both DNA strands at the same side. Okay, so this one is the vector DNA, another one is the foreign DNA. So EcoRI cuts the DNA between the bases G and C. Between the bases, so between the bases G and C, right? So no, GNA. So GNA only when the sequence GAATTC is present in the DNA. Okay. Now you see in the EcoRI acting on both vector DNA and then foreign DNA, so it forms the sticky end. So this forming a sticky ends, right? So this forms a sticky end. Okay, now DNA fragments join at sticky ends. So DNA fragments, these are joined at sticky ends. Now sticky ends, you see, the DNA fragments join at sticky ends, you see, they form the recombinant DNA. Now this is it. So this is the process of formation of recombinant DNA. So how this is formed? Now another thing is that GAATTC, you see in the 5' to 3', 5' to 3', this is the palindromic sequence. Now another diagram showing diagrammatic representation of recombinant DNA technology. You see there is foreign DNA, this is vector DNA, that is plasmid. So same restriction enzyme, you remember that same restriction acting on foreign DNA, also vector DNA. So same restriction enzyme cutting both foreign DNA and vector DNA at specific points so that they produce the same kind of sticky ends, so that they can ligase easily after the cutting. So ligase is join foreign DNA to plasmid. You see this taken the foreign DNA into this gap, so it fills and there is the plasmid formation, then transformation. Now transformation into the cell. So some cells get it and some do not. So non-transformation and transformation. So this is it. So this is the diagrammatic representation of recombinant DNA technology. Now there is separation and isolation of DNA fragments. The process is gel electrophoresis. See in the diagram a typical agar gel electrophoresis showing migration of undigested lane 1. So at lane 1 there are undigested, that is not used by the enzyme. So now other 2 3 4 lanes, they are digested set of DNA fragments. They are having the DNA fragments in one lane, there is no DNA fragment. So you see on near the it is the largest one and further they are the smallest DNA. So they are the DNA bands. So in the cloning vectors, you see cloning vectors. Now cloning vectors, what are the cloning vectors? That clones and vectors like this type of vehicle, like the malarial parasite plasmodium that carries the sporoid. So it kind of cloning vectors. Now they have some properties. Now what it mean to be a vector? So first it needs to be the origin of replication, so ORI. Then second one is selectable marker, third one is cloning sites. So in the first one, and fourth is the vectors for cloning genes in plants and animals. Okay, so vectors for cloning genes in plants and animals. So there is four things. So first one is origin of replication, ORI, that replicates. Okay, now a selectable marker, there is procedure, that is a transformation. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistant to antibiotics such as ampicillin, chloramphenicol, or tetracycline kanamycin are considerable useful selectable markers for E. coli, the normal E. coli. Okay, so this is selectable marker. They are showing the transformation. Then cloning sites. There are recognition sites for the commonly used restriction enzyme. So very few, preferably single recognition sites. So these are the cloning sites should be present in the vectors. Now there are like the ligation, and there is antibiotic resistant genes. The ligation of RNDNA is carried out at restriction site present in one of the two antibiotic resistant genes. So see in the diagram E. coli showing